Hello everyone, this is Harshvati and I'm back with another session on the genetics, and today we will be going to cover the topics like the chromosomal theory of inheritance and linkage. Okay, so I hope that you have watched the recordings of the last class, and this class also you are present. Now this class is kind of very much important because here we are going to discuss the linkage, and you like 90% of the students leave that Bhyanaksa diagram from the NCRT like this and this. You guys don't understand like what's going on with these diagrams, so don't worry, today we are going to figure it out, everything about these diagrams. Okay, chalo, so let's quickly start this session. First of all, let's talk about the chromosomal theory of inheritance. Chromosomal theory of chromosomal theory of inheritance. Inheritance. Sir, what is the chromosomal theory of inheritance? So to be very honest, it's nothing new. Just in this, they have compared genes and the chromosomes. That's all. Okay. So this theory of inheritance was given by what? The scientist which gave these theory was Boveri and Sutton. Okay, Sutton and Boveri and Boveri. Dates you don't have to remember, but I know the date, so that's why I'm going to write it. So the date was 1902, which is the year basically. Okay, so Sutton and Boveri were the scientists who gave these chromosomal theory of inheritance. And see what they have done. They have made a comparison between the Mendel and the Mendel experiments, Mendel conclusions, Mendel factors or genes between the chromosomes. Okay, so what happened during the Mendel when the Mendel was having the experiments? So at that time, major thing was the communication gap, and due to the communication gap also, you know that his experiments was not able to publish during his time. After his death, the experiment was published. So when these people, the Sutton and Boveri, were checking out, so they saw that there are so many things which Mendel has already said, and they are kind of related to chromosomes only. So kind of they made the comparison between the gene and chromosome. Okay, clear? So according to us, gene is what? Gene is present in the form of pair, yes or no? Yes, obviously gene is present in a form of pair. So yes, let's write here gene, and then in the second column let's have the chromosome. Okay, so now we know that gene is present in the form of pairs, present in form of pair. You know the drill, right? If you have doubts, comment section is your place where you can comment your doubts. Okay, what about the chromosomes? Yes, we know that homologous chromosomes are also present in pairs, yes or no? Homologous chromosomes homologous chromosomes are present in pairs. Okay, great, sir. This though we know. Next, what? Second thing is that genes are what? Genes are present in the form of alleles, as we say, they are present in the form of pair, and both during the gamete formation, what happens? These alleles get separated, yes or no? Just like this. See, if I'm talking about a gene, so here we have capital A, right? And this is a gene, and these are their alleles. Okay, so gene occurs in the pairs, right? And this is what? This is one allele, and this is one allele. If I compare this with the chromosome, so this is the chromosome, homologous chromosome. So I can say the chromosomes are what? The chromosomes are in pairs. And now they said that during the segregation, gene pair or allele segregates during the gamete formation. Yes or no? Okay, it's very true. See, if you are making the gametes from capital A and capital A, so what is going to happen? One gamete will have capital A, the other gamete will have capital A. In a similar manner, if I talk about the chromosomes, so in chromosomes also, when the gamete is made, one chromosomes goes in one gamete, the other chromosomes goes in the other gamete. If you're not able to understand this, let us understand this like this. See, you have X chromosomes and Y chromosomes. During the gamete formation, X goes in one gamete, Y goes in one gamete, right? If I talk about this, so we talked about the chromosome, okay? Whatever you are doing, pay attention here. If you're going to pay attention here, then you're going to understand the toughest part of genetics. Next we have what? If I talk about the with respect to gene, so let's suppose you have a gene which is representing capital A and small a, okay? Okay, clear? So this capital A is what? This capital A and small a is what? Gene pair, right? If they have to form the gamete, so one gamete is going to have capital A and the other gamete will going to have small a, right? So this is the second point, that during gamete formation, alleles are what? Or genes what? Gene pair segregates. Yes or no? Gene pair segregates. Clear, sir? No doubts. Similarly, in chromosomes also, homologous chromosomes, like the chromosomes, segregate during the meiosis. Yes or no? During the meiosis, when meiosis is happening, so chromosomes are what? The chromosomes are separating from each other during the gamete formation. So I can say that chromosomes separates during meiosis, right? So here Mendel was saying these things and Sutton and Boveri also said these things, and they compared both of the things. So basically gene is representing what? Gene is a kind of a chromosome only. Whatever the things are true for gene, it is true for the chromosomes also, okay? Then it said what? Independent assortment. Mendel said genes are independently assorted. Yes, obviously in this dihybrid cross proved this thing. Here also they also said that chromosomes are independently assorted. It's not like that our eye color and skin color is linked. If I have a black eye color, sir, you will have a black skin also. No, it's not linked. Clear? Okay, so they are what? They are separated independently. They are inherited independently. Okay, so this was the thing which was made by what? Which was made by the Sutton and Boveri, and they gave this chromosomal theory of inheritance. Okay, any doubts still here? Anyone? Let me know in the comment section. Now, let's come to this diagram. Let me make this diagram little bit bigger. See, I think so now it's little bit clearly visible, right? Chalo, great. See, if I talk about case 1, this is case 1 and this is case 2. Okay, I'll explain you this diagram. Just see here, look carefully. In this case, what's happening? In this case, there are two chromosomes, okay? Chromosome number 1, let's suppose, and this is the chromosome number 2. Now, in meiosis 1 of the anaphase, this is the condition. Yes or no? In meiosis 1 of anaphase, what is it? This we are having the condition. What's happening, sir, here? In anaphase, we have what? We have the homologous chromosomes. And what happens? The homologous pairs of chromosomes get separated in the meiosis 1, right? But in meiosis 2, what happens? In meiosis 2, these sister chromatids are being separated. Here what happened? Here you remember, if I talk about the cell cycle, if you have any doubts in these terms, now go and watch the cell cycle. Okay, that will be very helpful for you. So what's happening here, sir? Here the bivalent which were formed, the bivalent or the tetrad which were formed, they are separating with each other. In meiosis 2, what is going to happen? In meiosis 2, the homologous pair will be, the homologous chromosome, the chromatids, the sister chromatids of homologous chromosomes will be separated. Okay, or I should say the sister chromatids of the chromosomes will be separated. Okay, sister chromatids sister chromatids will be separated, will be separated. Okay, so here you can see we have some combination, like from here it is separated, from here also it is separated. Then what? Then we have the same kind of combination, this orange with green, orange with green. Okay, again yellow with red, yellow with red. We have these combinations, yes or no? Yes, see, in parent also we had what? Orange with green, yellow with red. We have these same combination, yes or no? Yes sir. Clear? Great. If you didn't understood this thing, again I'll help you to gain some learning here. See what is happening basically. If suppose you have, if suppose you have the chromosome in S phase, okay, a chromosome in S phase, it is having what? It is having a 4C DNA content, right? How is that? See here. How, what, just remember how many chromatids you can see here. Okay, whatever the chromatids you can see, that is the number of the DNA content. Okay, that is the DNA content. So here what's happening? Here this particular DNA, this particular thing is having what? It is having 4C content. Okay, 4C content, double ho gaya content. Okay, and but the chromatids is what? The chromatids are attached to each other. So I can say the ploidy is 2N only. Again I am saying, if you are not very much clear about this thing, go and watch the cell cycle. In that there is, this thing is very very clear. Okay, now what is going to happen? There will be one more pair homologous है, तो इसका एक और pair तो होगा. So let's make one more pair. Now in during the meiosis 1, during the meiosis 1, what is going to happen? They are going to form bivalent or tetrad structure, yes or no? They are going to form bivalent or tetrad structure like this. Yes, meiosis 1k time पे what is going to happen? They are going to form this bivalent or tetrad structure, right? Let me write here bivalent or tetrad. Clear? Now during the meiosis 2, during the anaphase 1, anaphase 1, what's going to happen? These will be separated. Okay, they will be separated. After crossing over will also takes place, right? So after crossing over, some of the parts of these pink chromosomes will be there in some of the parts of the yellow chromosomes, and similarly since they had crossing over, so their genetic material will be interchanged, and in anaphase this bivalent is totally separated. Now what happens here in anaphase 2? In anaphase 2, these chromosomes having the chromatids, having the common centromere, they also get separated, right? They also get separated. So see here, now what will happen? We are getting these 4 chromosomes, they are also separated. Again guys, if any problem is there in understanding this thing, go and watch the cell cycle lecture. I'm telling it again, stop the video first, go and watch the cell cycle. I'll give you the link in the description, okay? And I'll also put the timestamps key from where to where you have to watch, okay? But make sure you watch it, because without that this will not be clear, and the whole cycle, cell cycle, I can't explain in this part, right? So see, this is happening. Here also something similar is happening. In meiosis 1, the bivalent and the tetrad was formed. The tetrads are now separated, and the chromosome, the chromatids attached to the same centromere are also separated here. Okay, so we are getting 4 types of gametes. Here the combination was what? Here the combination was the orange with green and yellow with red. Here the combination is what? Here the combination is yellow with green and orange with red. Yes or no? Yes. So can you see there is independent assortment. Like it's not like that ki orange will go with green only. Orange can also go with red. Yes or no? Okay. Clear to everyone? Yes. Is it so? Okay, great. So these were the things which were explained by the Sutton and Boveri, but they couldn't give the experimental proof. To give the experimental proof, what we have to do, we have to call the Th Morgan. Okay, to give the experimental truth, we have to call the Th Morgan. Guys, just give me a second. Okay. Yes, now I am back. So to give the experimental proof, who came? Th Morgan. You remember Th Morgan? We call Th Morgan a very great person, father of something, right? Who was he? Like, what was the title? What was the title? The title was, guys, the title was the father of experimental genetics. Father of experimental. Wow, sir, you're writing the spelling of experimental wrong. Okay, okay, ठीक है. Father of experimental genetics, right? Okay, chalo, great. Now, if I talk about this thing, which is the father of experimental genetics, and let's check what actually did Th Morgan do, like how he proved the chromosomal theory of inheritance and how they have got the term linkage and all those things. So everything we are going to discuss. Okay, so first of all, Th Morgan did what? Th Morgan took a plant, sorry, not plant, I'm so sorry, took an organism whose name is Drosophila, that is fruit fly. Okay, so he did the experiment, and like Mendel was like P, 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 only P, only P. So Th Morgan was like, no, 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 I'm not gonna do the experiment on P, rather I should do the experiment on some other organisms like the Drosophila. Okay, so experimental organism. Here, the experimental organism is what? It is fruit fly, or I should say Drosophila. Okay, fruit fly, or I should say Drosophila. Clear? Now you know the whole name, Drosophila melanogaster. You write it down. Take it. Why, sir? Why we have taken fruit fly? Why? The question is why he has taken, why this person has taken fruit fly only. So see, there can be the many reasons, but some of the reasons are what? Why? Why he has taken? Because he liked fruit fly. No. In genetics, if you like or not, it doesn't depend on your preference. Okay, this might be some very logic. So see, food medium can be easily available. Food can be easily available. Like see, uh, fruit flies, they need sugar kind of thing. Okay, so either you can give bananas, like mash the banana and create the medium. There they can grow. They can grow in sugar water also. So food can be easily made available. We don't have to do much things there. For example, banana medium, right? And here the progeny size is again very large progeny. See, large progeny matters a lot. As I gave you the example, let's suppose that let's suppose there are four students. I went and I interviewed four students, and all the four students said that no sir, I got 300 marks in it. Then the other student said, the second student said I got 200 marks, and then the third student said I got 150 marks, and then the fourth student said, sir, I got marks in negative, just for an example. So I just interviewed four students in a room, inside a room, and I don't know about any other students, and I don't know about the any other thing. So I can give you a result on that basis that the maximum marks was scored in Neet 2024 was 300. But actually it's not. My thing is wrong. Why? Because my progeny size was so low that there is kind of, so many problems. They like, if my progeny size is so low, then I can give you the bad results or the wrong results, like I just gave you. Maybe they outside my room, there are so many students which have scored 720 maybe, right? But I decided with my small experimental students, or the small experimental organism, I decided that okay, this is 300. So less number of progeny makes results, what makes results little bit confusing, makes results little bit wrong. So this is why large progeny is necessary. Okay, so now their lifespan is short. Okay, their lifespan is shorter lifespan. If the lifespan is shorter, guys, then what? Then say it will be easy, na? It will be easy to have the next generation, fast next generation, and if the next generation is produced fast, then what? Easy to study the characters that how the one character is going into the other generations. Clear? Okay, sir. Now, let's come towards the things like what did Mendel did. So here again, just like the P, contrasting characters were also available. So Mendel chose some of the contrasting character. Let's see. Okay, one more thing. Here selfing was not possible. Okay, selfing was not possible because male and female are present in different bodies. Now see, let's talk about the crosses which were done by the, did I said Mendel? Oh my god, Morgan. Okay, so let's see cross 1. So in cross 1, character, the number one character which was chosen by Morgan was body color. Okay, and then the body color was basically of two types. One is brown, the other one is yellow. Now here there is little bit of, changes in the representation. So although the brown is dominant and yellow is recessive, but we're not going to write brown as capital B and yellow as small b. Rather, what we're going to write here, Morgan, this thing, we're going to write the letter of those which is recessive, and if we have to show the dominant character, we are going to use the same letter for which we use for the recessive character, but we will add a plus sign. Okay, now after this, Mendel chose the second character also. In cross 1, he chose the second character as well. And what is the second character? See, the second character was the eye color. The second character was eye color. Okay, and here there are two different types of eye color, yes or no? One is the red eye, and the recessive was what? The recessive was the white eye. The recessive was the white eye. Yes or no? Clear? Any doubts still here? Any doubts still here? Let me know in the comment section. If you have any doubts, please let me know in the comment section. No doubts? No doubts, anyone? Great. Great. If you have no doubts, don't worry then. Further also, you won't have any doubts. Chalo. Now, let's talk about what? Let's talk about the cross. So see, whatever the cross in NCERT is made, I am going to make this one cross A. Okay, and if I make this cross A, this cross B, you are going to understand by yourself. Okay? Now, but see, unless and until you're not going to practice cross by yourself, things will not get clear. So whenever you are revising this chapter or whenever you are making notes, try to make the cross by yourself. Okay, see here. So let's take yellow female with white flower first. Okay, the female is having X and X chromosomes. So I am just representing the X chromosome like this. See, let's represent the X chromosome with the line and Y chromosome with this one, like this. Okay, so I'm saying that this female is what? This female is yellow and white. This female is yellow and white. Okay, so while the male is what? Brown and red. brown and red. Clear? So if I'm taking the female is yellow and white, so what kind of genotypes they can have? So for yellow, they can have capital Y, sorry, small y, okay? Because what's Y? See here yellow was recessive, okay? Now, what about the color white, sir? So the white is what? The white is the color of I. So the color of I here is also recessive. But what in the case of this male? So in case of male, we have what? We have brown color. So I can write here Y plus, yes or no? And W plus. Now see, this is the X chromosome and this is the X chromosome. This one is the X chromosome and this turned one is the Y chromosome. Now just imagine there is no X chromosome, like only one X chromosome is there in the males, right? So these genes are only and only present in X chromosomes. They are not present in Y chromosomes. So I have not made any genes in Y chromosomes. Why, sir? Because भाई ये X chromosome पर ही present है. They are not present in Y chromosome. So I just can't make here Y and W, right? Because Y chromosome doesn't have these genes. Okay, clear. Now F1 generation. Let's make the F1 generation. So with the help of the diagram only, let's understand. After that, I'll explain this thing with the help of the Punnett square also, so that you will be able to understand this thing very completely. So if I talk about the F1 generation, so we can have Y plus W plus, or we can have YW combination, and we can have Y and W combination. Okay, sir, how we got this? See here, I'll explain you. Okay, don't worry, I will definitely explain you. Now let us represent this thing with the another way, okay? So let's have the X chromosome for the XX and for the female and XY for the male, right? So see, X chromosome we have Y and W, right? Let's write here. So X chromosome we have YW, and again with the X chromosome we have YW. Yes, sir. In the YX chromosome for the male we have Y plus, and we have red, okay, W plus. But on the Y chromosome there will be no genes for this body color and the eye color, right? Because it's Y chromosomes and Y chromosome doesn't have this gene. Now we are crossing it. So how many types of gametes you are gonna get here? So we are going to get one type of gamete, which is XYW. And from here, sir, we are going to get a gamete which will be X, Y positive, W positive and Y. Clear? Yes or no? Say it. Yes? Clear? Any doubts? Any doubts anywhere? Let me know fast. Now, let's make the Punnett square for this. Let us make the Punnett square here. How we are going to make the Punnett square? See. Let's talk about the female gamete. So in female, what we are going to get? We are getting total we are getting two types of gametes, two gametes, but same type. So let's write here XYW, XYW. And in males, what we are getting? We are getting one type of gamete, which is X, Y plus W plus. Okay. X, Y plus W plus, and what we are getting? And we are getting, sir, Y. Okay. Now let's make the Punnett square here. Let's make the Punnett square. I know my Punnett square is kind of very bad right now, but thoda sa you have to manage. Okay. Chalo. So first this and this. So X, YW and X, Y plus W plus. Yes or no? Y plus W plus. Then let's have this and this. So X, YW, and then we're having X, Y plus W plus. Then what? This and this. So see, X, YW y, and then again this and this. So x, yw and y. So this is the combination, this is the thing which we are getting. And see, we have written this thing only, na yahan pe to wahi likha hai humne. In the diagram, we have written this thing only, y plus w plus yw. See, females, yw, y plus w plus yw, y plus w plus. Yes or no? We are getting this only. Yes. And here, we are getting yw. We are getting yw for the males. Yes or no? Now understood what they are representing. They are just representing the genotypes which are they are producing, right? Nothing else. It's very easy. It's very easy. I'll try to show you this diagram also. 1 second. If you are able to see, I'll zoom it more. Can I zoom it more? Yes, see here. Just focus on the cross A. Just focus on the cross A. This was what? This was YW. Yes or no? YW. We have taken that only. We have taken that only. See? YW, 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 YW. Yes or no? And then what, sir? Then we have Y plus W plus. Yes. And here we had the Y chromosome. So nothing is present in Y chromosomes. Then we had the crossing over. So in crossing over, can you see? Can you check here? We are getting what? We are getting XYW. We are getting this combination XYW and XY plus W plus. So can you see the same combination here also? Can you see the same combination here also? Check here. Can you have this? YW, Y plus W plus, and male में भी YW आ रही है, right? Okay, understood? Now understood easily? Chalo. Now what will happen next, sir? See, we can't do selfing here, right? We can't do selfing in this case. But what we're doing, whatever the progenies are produced in F1 generation, we are what? We are allowing them to mate. Okay. So, in F2 generation, the parents, if we are considering the F2 generation, so the parents will be what? The parents will be F1 generation, right? So Y plus W plus we are having, and then what we are having? We are having Y and W. And this will be what? This will be crossed with YW and this one. Yes, because this was what you are getting in the F1 generation. This was the thing which you were getting in the F1 generation. Okay, great, sir. Now what? Now see. Again, let's make the Punnett square. Punnett square से ही समझते हम. Okay, Punnett square से ही समझते. Let's make that. So the gametes will be. Can I write this like this also? X Y plus W plus X YW. If you're having trouble in understanding, let's make it in the terms of X and Y. Here we have X, YW and Y. Yes. So how many types of gametes they are gonna make? Here we are going to get one gamete which is X, Y plus W plus, and this gamete which is X, YW from the female part. Yes or no? And if I talk about the male thing, so in the male thing, what gametes we are going to get? Sir, one is this and one is this. So one gamete X, YW, X, YW and Y. Okay, great, sir. Great. Now let's make the Punnett square. See here, very carefully see here. Now this X is going to get fused with this. So what we are going to get? X, Y plus W plus and X, YW. This X going to fuse with Y. So we are going to get X, Y plus W plus Y. Similarly, this with this. So we are going to get X, YW, X, YW. And this with this. So X, YW and Y. Yes or no? Okay. Yes, yes, sir. Great. Any doubts right now? Any doubts right now? No doubts, sir. Now let's make this with the help of the Punnett square, whatever we have got, let's make this in the form of this thing, okay, in which the NCERT is given. Or let's not make it, it's present there. One second, I'll search the diagram where it is, where it has gone. Yeah, see this one. I'll copy and paste to this. Oh, what happened? Are you angry on me, my dear image? See, let me do one thing. Let me show you this only, okay? So this was what? First of all, let us understand. This was what? This was F1 generation. Yes. Now we have the F2 generation, okay? Now we have the F2 generation. So I'm just showing you the pic of F2 generation, okay? See, I think so it's visible properly. So here what's happening? Here, can you see this? This is y plus and w plus, and this is y plus and w. So can you, let's check, are we getting these type of things or not? See, where it is, yeah, x y plus w plus, x y plus w plus, and x y w. Are you able to get it? X y plus w plus and x y w, y plus w plus and x y w. See, we have got this, right? Then what we have? X y plus w plus and y. So x y plus w plus and y, x y plus w plus and y. Yes, this we have. Yes or no? Y plus w plus and y. So in this manner, this cross is made. Okay, in this manner, the whole cross is made. Now, sir, what are we trying to check here? See, in this thing, Mendel said that, there are combinations. What combinations? Not Mendel, Morgan. Morgan observed that, here Morgan observed that he has taken what? He has taken parental type. Okay, the parent was what? The parent were yellow white, and brown red. Yes or no? The parent were yellow white and brown red. Here, where you can find yellow white and brown red? See, for the yellow white, we have what? We have y plus w plus. For the brown red one, here we have. Okay, now see, again for the y plus w plus, let's search y plus and w plus. This we have, right? Okay, now y plus w plus, again do we have? No. So yellow, brown and red we have seen two. Yellow and white, let's search about yellow and white. So can we have this as yellow and white? 1. And can we have this as yellow and white? 2. Yes or no? Yes, sir, we can have. Okay, great. Till now, no issues. They are what? They are the parental types. So now Mendel said that there are some new combinations also. What kind of new combinations? One is the y plus w. Like, see, in parents we didn't have y plus w, right? In parents we didn't had y plus w. See, in parents, do we have y plus w? No, we had yw or y plus w plus. So here what had happened? Here something has happened which has created a new combination. Yes or no? Which has created a new combination. 1 second, I'll go back to that slide. Just give me 1 second. What happened here? Yes, see here. So in case of some, we had what? We have the new combination. Yes or no? Yes, sir. but the recombination here is what is only 1.3%, while the parental type is 98.7%. Okay, great. So new combinations are formed, but how much? Just 1.3 percentage. And here parental type was 98.7. So if I ask you this, so you will be like, yes sir, the yellow wants to stay with white only and the brown wants to stay with the red only. And Morgan observed this, and Morgan said that this is just because that the genes are very close to each other, so they want to be linked, they are cross linked with each other. Okay, they are cross linked with each other. So this is why the new parental type is new recombination is not able to be produced in more amount. Okay, now he took the second cross, the other cross which we have in the NCRT. Yes or no? Yes. See, so if I talk about how did the new combination formed, if this is the doubt, see, I'll answer you. See, what can happen now? See, we know that in fecitine crossing over takes place, right? So let's suppose we have this. Here we have Y plus and W plus. Okay. And then we have what? We have Y and W. So crossing over takes place. That means what? That means here there will be the endonuclease. First of all, the enzyme will be there, endonuclease and nuclease SE. Okay, so this endonuclease will cut this chromosome like this, the genes like this, Y plus W plus, and then we will be having Y and W. Okay, and now what's now they are cross-linked, the crossing over is happening. So during the crossing over, we have the again the another enzyme which is the DNA ligase, and they are formed. The new combination are formed like this. Okay, clear? This is how the new combination is formed. Now it's clear how the new combination is being formed. Yes? Okay, great. Now, after this, let's come to the second cross of the Morgan. So in second cross, he took another two characters. Same Drosophila में ही हो रहा है, but he took different characters now. He took wing size. One character is wing size, and it represents normal, like normal was dominant, normal wing size was dominant, and then we have the miniature which is recessive. Okay, so this was dominant and this is recessive. So now here let's represent it with wing size as W plus and eye color as, sorry, wing size as M plus and eye color to as it is we were representing. So here in cross to one character was what? One character was wing color, wing size, which is in which miniature is dominant. So let's write M, and normal was, sorry, miniature was recessive. So let's write small n, and normal was what? Normal was dominant. So M positive. And the second character which they had was eye color. And in eye color, what they had? Just like the previous one, red and white. Okay, so here we have W, and for red we have W positive. Yes or no? Yes sir. Any doubts? See, W positive and W. Okay, so I have to become, tongue tilt here and there so that you guys can see the video properly. Okay, remember that this is a very important thing which we are learning. If you feel that you are not clear with what's happening, if you feel that you are not clear with the, one time it may happen that your concepts are not getting it into your brain, so rewatch this lecture again and again. Like if you have any confusions, I can bet you that you will able to understand everything. Okay, you are able to understand everything. One second. Chalo. Now let's move on. So when the cross was conducted between these two characters, so it was observed that here these two characters are what? See, these two characters are present very far from each other. These two characters are basically present far from each other. See, can you see this? The distance between these two alleles or these two genes are very far. Yes, as compared to this one. So in this case, the chances of recombination increased. Okay, so here when the cross was done, which was parental type, so we get 62.8% only, and recombinant we get 37.2%. And in this case, recombinant we got 37.2%. Okay, so now figure it this crossed by yourself, the second cross by yourself, just like we did with the first cross. Okay, sorry. If you are not able to understand the second cross, let me know in the comment section. I'll explain it. But I want you to first of all try by yourself. Don't just go and search for the another YouTube videos, because in that case you are not going to learn it. It totally depends on you. You want to, do the things with you by yourself to understand it easily, or just go on another video and watch. Sir ने बोला खुद से करने को, तो छोड़ो, let's do it by some other videos. No, you have to practice it, because you have to learn it. Okay, clear? Chalo, great. So I am leaving this cross on you. I have discussed everything. The main thing I have discussed, just how this is coming, W and M, how it's coming, what is W plus and M plus, how it's coming. You have to do it. Okay, chalo. So two things Morgan said, that he said that if the genes are very much closer, they want to be with each other, and if genes are far from each other, they are able to produce the new phenotype. Okay, clear? So now Morgan said that this is nothing but the linkage. So sir, can we define linkage? Yes, definitely we can define linkage. Yes or no? This is the linkage only, right? Whatever the things we were doing till now is basically what? It's basically the linkage only. So let us define. So can I say that linkage, this phenomena, what we are seeing was linkage. In first case, the parental type was very high. The recombination was very, very low, right? So we can say in these cases linkage can be seen. So linkage is defined. Linkage is the tendency of two different genes to remain same, together during gamete formation. Okay, right? So the separation is not possible. Like here, let's suppose there are two genes A and B, so they want to be together. Okay, they want to be together. So when the gametes will be formed, they will not get separated. In the gametes also they will be together. So this is the linkage which we are able to see in the cross 1 of T.H. Morgan. Here what was happening? The parental new combination was not able to form. New combination was very, very less, just the 1.3%. So that means what? That means this brown is linked with red and yellow is linked with white. When they will produce the gamete, yellow and white will go together and brown and red will go together. Okay, clear. But in cross 2, Morgan observed that if we increase the length, sorry, if we increase the distance between what? If we increase the distance between the what should I say? The distance between the genes, then the linkage will be what? Linkage is reduced. So can I say that strength of linkage is inversely proportional to distance between genes? Yes or no? If distance between genes is more, then strength of linkage will be less. Clear? So I can say if more distance between the genes, if more distance between the genes, then what is happening? Then sir, chances of what? Chances of recombination or formation of recombinant increases. Recombinant increases. Yes or no? Yes sir, great. So see, what are the factors which are affecting linkage? Let's write that. Factors affecting linkage. So it was very easy, na? And you were like, sir, we are not able to understand this diagram. Make the Punnett square. You will be able to understand it very clearly. Okay. For the second diagram, you know that drill. If you are not able to understand, make the Punnett square first, and then again, if then also you're not able to understand, I will publish a small video for that thing. Okay, don't worry, just let me know in the comment section in both the cases. Because see, if you're not gonna communicate with me, then how I'm going to know that what issues you are facing, right? So if I'm gonna say distance is more, so crossing over linkage will be what? Linkage will be less. Yes or no? If distance is more, then I'll say linkage is less. Next what? See, age also. If age is more, then chances of linkage is increased. And this is the reason why we say that couples should not wait for a long time to have a baby, because as the age is going to increase, the linkage is going to be increased, and it can create some problem. Okay, now temperature. If you increase the temperature, linkage will be decreased. Okay, next we have what? X-rays. If I'm going to give X-rays, then linkage will decrease. Okay, next what? Next is sex, gender. What about it? See, in if I talk about sex, so male Drosophila, there is no linkage. In male Drosophila, no linkage. Why, sir? See, I'm gonna say you, because in male Drosophila, they don't have the other X chromosome, right? Only and only one X chromosome is present. So there will be no linkage. See, is there be any linkage? No, there is nothing. There present ही नहीं है नहीं, Y chromosome है इनके पास. तो लिंकेज होने के लिए दो एक्स क्रोमोसोम तो चाहिए, तब तो ये दूसरे के साथ लिंक होंगे. सो व्हाट इज गोइंग टू हैपन? सॉरी, आई एम सो सॉरी, देयर विल बी नो क्रॉसिंग ओवर. ओके, there will be no crossing over. I used the wrong term here. There will be no crossing over. Yes or no? Because for crossing over to happen, there must be something present here and here. So it's Y chromosome, and in Y chromosome we don't have the characters for the eye color and the skin color for the Drosophila, right? So in male Drosophila, there will be no crossing over. So 100% the thing will be what? The thing will be here will be linked. Okay, so W positive and Y positive are linked with each other, because no crossing over can take place. Clear to everyone? Okay. Now, One more thing which hampers the or which affects the recombination is position of gene. If gene is present very close to centromere and telomere, then chances of crossing over becomes less. The chances of linkage becomes high. Okay, clear. Position of position of genes. Position of genes. So let's suppose if the gene is present near to the centromere, like here. So here what is going to happen? Low chances of crossing over. Okay, less crossing over, and crossing over is less. So what is going to happen? Linkage can be made. Okay, linkage will be high. In a similar manner, if the genes are present in the telomeres also, which is telomere is what? Telomere is the point which is the terminals of chromosomes. Okay, then also chances of recombination is very, very less. Chances of linkage is very, very high. Terminals of chromosome. So in both the cases, what's happening? In both the cases, the linkage is high. Linkage is high. Okay, clear to everyone? Yes, sir. Now just 10 to 15 more minutes and then I'll finish off this particular topic. Okay, and after this I'll give you some of the videos, like a very short video. Okay, a very short video tomorrow, in which we will solve some of the questions related to linkage and all those things. And after that, in this chapter we will be left with sex determination and sexual in sex dependence inheritance. So both of the thing I will be doing live lectures. Okay, in dono ka let me take the live lecture. So live lecture will be continued again from the 26th of October. So till then, you revise the NCRT till linkage. After linkage, I'll give you the live lectures, and in the between, let's complete reproductive health. Okay, so the plan is like on 24th and 25th, you will complete reproductive health. Okay, and this video you might be watching on 22nd of October. Sorry, this video you are watching on 21st of October, and then you are going to get one day off, which is Sunday, and on Monday we will practice some of the questions, and on Tuesday you do this reproduction, and then after this we will start with the sexual how to determine the sex of the organisms, and then we will define the chromosomal disorders and all those things. Okay, clear? So I want those sessions to be live, not recorded. So let's have those as live sessions only. Okay, in the middle we can complete the reproductive health. Chalo, now next is what? Next is linkage groups. Next is linkage groups. So what is linkage groups? See here, if I'm talking about the linkage group, so this is one chromosome. Okay, and they wants to, the genes on this chromosome wants to be inherited simultaneously, like no separations. Okay, so such type of genes will be called as what? Such type of genes will be called as linkage groups. Okay, clear? Okay, now number of linkage. Sir, how we can know that in a person how many linkage groups can be there? See, so here there is what? There is, let's suppose this is chromosome number 1. Okay, this is chromosome number 1. Okay, and since it is having one more pair, so we are thinking that okay, it might be diploid, right? It might be diploid. And if I talk about the linkage group here, so N is considered to be as linkage group. So this one can be considered as one linkage group. Okay, so it's very easy now. If you are a diploid organism, so whatever N you have, whatever the haploid content you have, that is your linkage group. Clear? Okay, because in one chromosome, whatever genes are present, they can be called as linkage group. Okay, so number of linkage group is what? Number of linkage group in a species. How we can find the number of linkage group in a species is generally equal to what? Is generally equal to the haploid content, to the haploid content of the of that species. Okay, so if I ask you that what is the linkage group for humans, what is the linkage group for humans, so you will be like, sir, this is 2n. Okay, n is equal to what? N is equal to 23. N is equal to what? So we can have 23 linkage groups. But see, in males this is something different. So we have what? We have 22. I'll write here. We have 22 chromosomes, and then we have one chromosome X and one chromosome Y. Because see, X and Y, okay, X and Y, they can't form. X is not having its own linkage group, right? X is just X, Y is just Y. Okay, here what's happening in female, we are having 22 plus X and X. So we can consider it as one linkage group, right? But here X and Y is present. So this is one linkage group and this is one linkage group. In females, both the X are same, right? The diploid content of X. But here what's happening? X and Y are different. So here X will form one linkage group and here Y will also form one linkage group. So 22, 23, 24. So in males, we are going to have 24 linkage groups. 24 linkage groups, right? Because they don't have their pairs. Now, in males, X is also not having a pair and Y is also not having a pair. X and Y is not pair. If X and Y would have been same, then we would have considered it as one linkage group. But X is also different, Y is also different. So both are the different linkage groups. Clear? Any doubts? Any doubts in the comment section? Let me know. Any doubts? No doubts? Okay. Great. If you don't have any doubts, then I will give you some examples and then we will over the class. Okay? So if I talk about the garden pea, number of chromosome is 14. N content is 7. Then linkage will be what? 7. Okay. This we are talking about the 2n, N and linkage. Again, next we have the other one, which is the human female and human male. Okay, human female, what we have? So we have 46, we have 23. But here what's happening? 22 plus X we are having, which is 23 linkage groups. But here what's we're having? 22 plus X plus Y, which is 22 plus 1, 23 plus 1, 24 linkage groups in human males. Okay, clear to everyone? Clear to everyone? Any doubts anywhere? Let me know in the comment section. Okay, any doubts? No doubts? Great. Okay, I'm ending this today's session, so be ready to watch the next session and mark your attendance. Okay, chalo, see you. Bye bye everyone. See you in the next class.